ICTP-SAIFR, IFT. It's nice to see so many familiar and new faces around. So my name is Ricardo. I will be one of the organizers of the school together with Pierre.
Hi, I'm Pierre.
So before we start with the opening lecture, we would like to give you a little bit of overview of what's going to happen in the next 2 weeks. So let's see if I remember how to do this, because it's been a while since I don't do it. So I will explain to you a little bit where you are, but I won't extend myself. If you are interested, you can ask around, or we can talk about that over coffee. So I'm going to skip the description of what this is. This is a theoretical physics institute that is part from UNESCO, part from ICTP. It's a very nice place with a lot of very good researchers and students who are also researchers. Going to the pragmatic things, in the coffee breaks, there will be coffee upstairs. Whenever there is coffee break in the schedule, coffee is there. Lunch is not organized by the school, but there are a lot of different lunch options around. Also, the campus has a cafeteria that have both made dishes, which is the direct translation from prato feito in Portuguese, and also it's nice. You can get some proteins with rice and beans and salad. That is probably the cheapest option. There are also cheap options in the back of the institute, and then there are more fancy options around there. We can self-organize for lunch. What else? There is the poster session that will be tomorrow, so please bring your posters tomorrow, those of you that have posters. And I think that's pretty much it. If I forgot something, I will tell you at some point. Enjoy the lectures, ask a lot of questions. We have the huge pleasure to have 4 very great lectures for these 2 weeks with us. So try to make the most out of them and the lectures. And Pierre is going to explain you a little bit more about the school itself.
Yes, hi. So this first week will be very busy with a lot of lectures. So we have first an opening lecture by Veronica Grieneisen from University of Cardiff. And then the 3 other lecturers, Johan van der Koppel, Erwin Frey, and Nigel Goldenfeld, will be alternating, giving the lectures, and each of them will also give a hands-on session that's a bit longer, and that is designed for you to get a bit to grasp with the practicalities of this of pattern formation. And then the second week will be more participative. So we'll have a full series of lectures by Veronica, and 3 lectures, so 1 by Ricardo and 2 by myself. A series of short talks in one session. And all the afternoons will be devoted to projects. And so these projects will be by small groups of 4 or 5 of you, trying to do simulations and implement and present some results about one or the other of the topics that we've seen during the week, the school. And at the end, the last day, there will be short presentations by about each of the projects. And with this, I think we're ready to start and delve into the beautiful problem of the mathematics of pattern formation. Veronica.
Working, 1, 2, 3, hello. Okay, so thank you so much for the organizers for inviting all of us here. This was a very nice opportunity to talk to an international body of students from physics. So the lecture that I'm going to give today is very pedagogical, and it's meant to be interactive, a bit to break the ice, and to think a little bit why we are doing modeling in biology. So I know that you guys, your origins come from physics, as do mine originally. But along my career, so I've done a lot of experiments. I've actually had lots of supervising students which are pure experimentalists in cell biology, sometimes in field experiments as well. And the question now is, what does it really look like doing, as a physicist, doing biology or doing mathematical biology, theoretical biology, systems biology, whatever you want to call it. Okay, so I think one question we have to ask ourselves is a little bit epistemological. So first of all, okay, what is it that we're going to do? Why do we do modeling for biology? And how is it done? So these things can be very different. And I'm pretty sure that you guys already have your experience and have some ideas of what it is that you consider is important for you in this field. And maybe at a certain moment we can address this question of what modeling is. I can tell you that when you start collaborating with biologists, what they consider is the objective of modeling might be vastly different than what you think it is. Okay, and I want to give a couple of case examples today. I think in the second week I will also have opportunity to show you a little bit more of some lectures which will be more pedagogical, more structured through these things about what we do in our lab. Just to tell you really quickly, so I'm very interested in pattern formation, but I'm also interested in how pattern formation can be robustly encoded. So why is it that development is always very nicely giving you the correct results, but also when is it plastic? So when does it listen to the environment and change its responses? And once you start understanding this conundrum between robustness and plasticity, you can also start looking at pattern formation over evolutionary timescales. So once you understand the mechanism, you can also start seeing how is it adapted, how is it changed, how is it harnessed for different species and for different instances. So that's something that you guys will see further on. Okay, so now when we talk about modeling, we have to realize that a lot of where we come from and what shaped our mind about science is this reductionist approach to science. And that's probably, I bet, what most of you have seen, right? And there's beauty and a very great strength in this, right? That is like, you take away all the noise and all the things which aren't important, you go to the core of the process, right? And sometimes actually, you do this even with the Gedanken experiments. You have to imagine this idealized situation, and then you can understand a process, right? So, quoting Sir Isaac Newton, truth is ever to be found in the simplicity, so quite a strong statement, and not in the multiplicity and confusion of things. Okay, now we will have a problem when we go to biology with this, yeah? Okay. But then again, it's not just physicists which would think like this. So this is Francis Crick, which for many people in biochemistry, would be their Isaac Newton. So he says the ultimate aim of the modern movement in biology is to explain all biology in terms of physics and chemistry. Okay. Now, you might think, oh, this is good for you, but actually what I'd like to advocate, and the other speakers will also be showing this, I think there's a new type of physics, a new way of approaching biology, okay? Which isn't just having to put it down to the basic chemical or chemical interactions. In fact, that is a distraction. So depending what you're looking at, and mostly the way life works, right? Yes, everything is made out of atoms. Everything in the end will be quantum mechanics. But those principles are insignificant to the processes that we're looking at, yeah? Explanatorily. So during these 2 weeks, I think one image I'd like you guys sometimes to think about, maybe in your lives, is this story that comes from the Middle East, from India, actually, Asia. So it's a fable, right? So they have this elephant, and these monks, which are blind, they go to the elephant and they're going to touch it, and they're going to touch a part of it, what they can touch. And they're very carefully studying, right? One is studying the tail, the texture, they're getting all the information they can. And then they come together, right? Bear in mind that they cannot see. And they will describe their experiences. So their experiences are all very correct, they're very honest, they have been observing very, very well, right, what they have been able to touch. But they will have very different accounts of what it is that they see, yeah? And so this is the issue in science, and we forget this a lot. So there is something out there, like the elephant in the room. There is something which is the truth, maybe, possibly, right? But we are limited. We always will be limited, either with the theories that we have or with the experimental interventions, the way we look at it, right? But also, we will be analyzing the elephant through different approaches. So during these 2 weeks, I think we'll be seeing lots of things passing by. We will be seeing dynamical systems, ordinary differential equations, partial differential equations for pattern formation, maybe some cellular automata, some stochastic modeling, agent-based modeling, right? Very different approaches. As you guys know, approaches will have their strength and they'll have their limits, their weaknesses, and it's very difficult to know now what can I say about a system which is beyond the formalism I'm using? Or what were the simplifications I've done in my elephant to try to understand a process? Was that a good simplification? Did it reduce it to a way that I could better understand, like the elephant? Or was it something where I'm just actually looking at a tail, right? And very importantly, when we start talking, when these, the story goes, the fable goes, that when these monks start talking to one another, there are different versions of the story, and say that they couldn't agree, right? Because they thought, you're lying, because I saw this, right? That they started having like, what reviewer 2 can do with your paper sometimes, right? Not because he's always against you. Sometimes he might just really be convinced that it's something else, because that's, the way they look at it. But so they fight, and they can't get out of that. And another thing, they actually do talk, and they're able to piece it together, right? And I hope that this is something that, as a community, interdisciplinary community, we can understand that we will have different backgrounds, we will have these different ways of viewing things, but that we can still understand each other when we talk, that we know what the other person did, what they didn't do, and that we can try that way to help one another collectively, to understand ultimately what we want to do, to understand something in biology better. Okay. Does it sound, yeah? Okay, so I use the word systems biology a lot. Again, it's one of those words that means a lot to some people, but also means nothing because there's so much that it can portray. But I just wanted to now start talking like historically about the idea of modeling in systems biology by another biologist, Dennis Noble. So he's actually someone who looks at the modeling of the whole heart system, so coming from biomedical. And he said already some time ago, systems biology is about putting together rather than taking apart. So this is not reductionist or not analytic. Integration rather than reduction. It requires that we develop ways of thinking about integration that are as rigorous. So we're not talking about flaky things like, oh, everything is there. No, we still have to be really rigorous about it as our reductionist program, but different. It means changing our philosophy in the full sense of the term, okay? So, and this is something which is very nicely said, but it's very difficult to understand, what are these new theories? What are these new ways of working? How do we actually formalize them, right? And I think that's why it's really important that people from physics enter here, but also with an open mind, that there is a lot of mess, but there are ways around the mess, okay? So when I say about mess, luckily, you guys will not be staring at things like this, right, too much in these 2 weeks. But this is sort of what's underlying biological processes. So we have these like hairballs, and if you go to a biological meeting, you'll see a lot of them. And they're forming even more interesting hairballs because now they can take cells at different locations, and you just look at connectivity between proteins, or which protein binds with what other protein, or genes. What gene is activating, suppressing other genes, and how do these things look like, right? And you can, in a high throughput way, you can actually collect type of like material like this. Okay, so here we have a couple of examples. So yeah, yeast transcription, you can just read it there. Protein-protein interactions, phosphorylation networks. So then the proteins act on one another to phosphorylate, to activate them. Metabolic networks, so how does the energy flow, right, get metabolized? They're very complicated networks. And genetic network based on if you take a gene out, what's happening? If you take another gene out, what's happening? Okay. So why are all these levels very important? Because they tell us something about something either being regulated on the transcription level, a gene being activated or not. But then when the proteins are there, they can also interact with one another, and they can also modify one another. So you have very different response scales. You have many layers. And you even would have, supposedly, the epigenetic layer, which is now going on top of that. Okay. And this is to understand why something is happening. Why does a cell divide? How does it divide? How do you respond to a different signal, right? So these are the, like circuitry, right? I open my computer and I look at the circuits, right? And very similar, people try to now figure out ways that they can look at topology of these networks, possibly, to try to not just stare at a mess, but try to simplify things, or look at look at the beauty of, well, maybe that we have hubs here. We can now look at these things and say there's a red module, a green module. It's a little bit like this. And then we can, again, isolate those sub-networks and say, OK, maybe we can reduce this to fewer links and nodes. So for example, for budding yeast, this would be like a quite concise network here of cell cycle. Like, how does that yeast divide, right? And when I say simple, even if you now put this into a Boolean approach where you say every node is a gene can be on and off, okay? So we have 2 possibilities for every node. We have 80 nodes. We have 2 to the 18 possibilities. But then the next state depends on the 2 to the 18 possibilities. So we have 2 to the 18 possibilities. So we have 10 to the 78,000 ways in which possibilities of the first state and the next state, only of this network. So it's very, and this is the most simple, this is the very simplified network, right? And the states in which it can be are basically endless, right? So, okay, but there are ways forward, and it's not that I'm giving you here a lecture, just a taste of what people in systems biology would consider is like one way out of this, right? So they look at the cell cycle division network, and now they do something, well, they're going to consider a Boolean logic, right? So things can activate and inhibit. So there'll be nots, like trues and falses. But one thing that you see here, this is a very consolidated experimental module. So we know that, so CTRA is inhibiting GCRA, which is activating CTRA, okay? Very nice. But for example, we start having, like in certain nodes, we have like multiple arrows that come in. And for that, sometimes it's very difficult to know, do those arrows work in conjunction? Do they work independently of one another? So for example, if we look here at DNAA, we have 2 green arrows entering, right? Does that mean that each node can activate it independently, or do they need to, for example, form a heterodimer to activate? So sometimes those things are actually not that well known. We just know activation, inhibition. So what people do here is then they do like they call this a stochastic Boolean network, which means that they synchronize from one time step to another. They don't synchronize. They just go through it in any type of sequence, right? From your state to your next state. And they're looking at all the possible combinations of these rules, which they know are experimentally there, but they don't know in which combinations they are needed. If you have ANDs, or if you have ORs, or you have XOR gates. Yeah? And so, just with this then, it means that you have about 500,000 type of possibilities. Yeah? And then they can take a look at what type of dynamical output do you get. So that's the behavior class. So for example, if you have a type of loop in which you still make decisions, because this is stochastic simulation, we have those double arrows. So we have that number of outputs in which it happens. We also have fixed points, so these are equilibrias. So we have 165,000 equilibrias. We have cycles coming. And then we have other situations where we can have 2 equilibrias. And this all depends on what specific rules are we using for combining interactions, positive and negatives. Okay, so this is basically, a long story. If you guys want to know a little bit more about this, but this is what some people would think is systems biology, right? Is you're trying to still have this meso complexity where you have enough that you can really say something about the system. You can do some type of analysis that you have a good handle on. You can look at attractors, limit cycles. Sometimes they get really messy, so these are the states in which they can go through. This is all for budding yeast. But what I want to tell you, it's very important now to realize that we always have to go back to the biology. For example, that number that represents like the first gene is on, the second is on, third is on, fourth is on, fifth is off. And sometimes you can say, okay, I know that state exists. It is an equilibrium. Okay? That's what the biology tells you. So now you can go back and you can constrain your network. You can say, well, if that is it, then I would actually have to update to get that to be one of the stable outputs, I would have to actually change my logic. Or I have to impose that as one of the logical rules. That's one thing you can do. Another thing people can do now is that they can say, okay, there's this hypothetical link here between CTRA and DNAA, let's say. okay? Is it there or not? Do you need it or not? Is it sufficient or not, right? So then we can start going through, these type of topologies and ask the question, what is happening with the dynamics of our output states? Yep. But you can do it in a more sensible way. You can make it into a truth logic way. We don't want to look at this jumble of outputs. We actually can now say, okay, either it is essential or not. If it is, we would get mixed phenotypes. If it's not, actually, then we need something else to be essential. So we can go through these conditions. And at that moment, it's really important to hook up with experimentalists and work together, because they're testable, clear ways of getting to know what's happening with that underlying scheme. Okay, so this is just a taste, like a case study about how it would be to do systems biology on that network scheme. Now, I myself am much more interested in this problem, right? In looking at how you come from an egg, so DNA instructions by themselves, almost by themselves, and how there's an unfolding, which isn't only genetics, it's not just transcription, it's not just translation, actually it's much more than that. There are biophysical processes. Now we have things which are diffusing, we have morphogen gradients, we have things reading out the morphogen gradients, we have cells, right? Cells that biophysically interact with one another, that move around, and all of these different levels, some of them which are more material, they interact to unleash the potential of what the DNA instructions are telling you. Because the DNA is not telling a cell, move to that place, move to that place. No, these are processes which emerge, and there's this self-organization happening, right? So there's much more complexity in the outfolding of this, structure of the bird, and the metabolism, the physiology, and its responses, than there is just if you can't get that information if you just look at the translation, transcription, and those hairy balls that I'm showing you, right? So then there's another part of science which is just thinking about that. Let's understand pattern formation. Let's understand the dynamics of the pattern formation. Let's understand how these patterns react within cells and across cells, okay? So that is something which is really important, and there's a big space here for physicists, for biophysics to work in. But even there, we have to interact with data. Okay, so this is just something I'd like to mention. And for example, this is some data from a PhD student of mine for some time ago. So this would be like epidermal cells on top of a leaf, okay, of a developing mutant of a plant. So it's interesting the things we get into when you start actually working with experiments, because you start looking at images and think, oh my gosh, this is, I have to segment things here. So what is it in the end of the day that this image was containing? Just a bunch of numbers giving you the intensity, right? So, there are lots of segmentation methods out there, but we actually developed one coming from physics, which is called the segmentation Potts model. So we use Potts model to be able to, with some biophysical constraints, to be able to find cells. And this ends up being a big part of sometimes what you might end up doing. So at the moment, this is a pipeline a postdoc of mine developed, in which we are looking at, together with a group of Stan Murray, in which we are actually looking at organoids of humans for cancer. But the problem is now, you can plate so many organoids, like thousands, and you can actually quantify them very quickly. You can image them very quickly. But it's not that easy to look at them and to know this is a normal organoid, this is different, or now I put a drug and this organoid has developed differently than that one. So first problem is to be able to segment it, to get cell characteristics out of that. But the other part now, the other problem, and this is still an open problem out there, is how do you extract properties that you can classify if something is going in a normal development or not, right? Now, a big answer for that, so this is a problem for example, cancer biology, but also here, for example, we were looking at data of the meristem of a flower, so very small stem cell niches of developing plants. So this is in 3D. So again, the segmentation has been applied, and here to the right, we also have staining of what location in the cell cycle are you as a cell, okay? And we're looking at this data because we look at it for wild-type plant, but we also look at mutant plants in which something has been modified in the way it fuels its shape and in the way it's able to divide. But the way we touch the elephant now is that basically we have a dynamical process. We can see every single cell. We can see what they're wanting to do. We can compare normal with altered. But you have to come with the rules. What are the rules which are guiding cells to normally divide, to know when to stop dividing? Come with a hypothesis, test the hypothesis. What are these hypotheses? Can we get a hypothesis out from the data extraction, right? And it's very difficult, like, because when I live in Britain, and when I look at people playing, I don't know, rugby is okay, there's this other game, cricket. I have no idea what they're doing, right? Because I don't know the rules. I look at them and I really try to, oh, is that the rule? You try to infer something from a behavior that you see. What's the underlying? And one way that we go about that very much is by looking at the topology, sometimes simplifying, stripping it down. So sometimes you can just think, okay, who are the cells neighboring? Is what a neighbor cell doing having an influence on yourself or not? That could be one approach. So for example, I'll show you how an approach like this with data has worked in this case study here. So again, we're looking at the top of a leaf. Yeah. The leaf is here at the bottom. This is actually a confocal microscope picture of this Arabidopsis leaf, which is growing. It's a mutant. It doesn't have the stomata. It doesn't really matter. But, there are lots of interesting questions here. First is that you have like a gradient of cell complexity. So cells, they start very like squarish, and they become really crazy shaped, right, as they grow. So there's a lot of cell polarity studies that we have with this system. But, because we were looking so much at the cell shape and cell polarity and how that works, we suddenly, actually, a student of mine said, let's just take a look at how they're dividing. Let's forget about we like it because it has those crazy shapes, but let's just look at the divisions. How does it do divisions? So for that, first of all, we had to there's a technical challenge. So we had these leaves growing for 9, 10 days, in which you had to image them during that whole process to catch divisions, for example. But we started to ask the question, what about the topology and the life history? So because the thing about life history, which is nice with plant cells, they can't move around. So depending on how they divide, you will see that afterwards in their topology. For example, down there, actually I have let's see if this works. Haha, okay? So for example, now think about the cells, think about the center of masses as these little, like these little nodes, right? So we have a graph here, right? And so this is how those cells, like 3 is connected to 1, 2, 5, 6, and 4, and it's sharing a cell wall with them. Now this cell number 3 is going to undergo a cell division, so it's going to create a new cell wall. Let's say it creates it in this way here between cell 1 and 5. So suddenly we're going to have, now after the division, we're going to see 2 new cells, a daughter cell and a mother cell. We like to call daughter and mother cells, quite cute. Okay, so here we have 3A and 3B. So what happens now is the topology has changed, right? So now, like, cell 1 is going to have an added neighbor, right? Cell 4 still has the same neighbor, but cell 3B and cell 3A have different number of neighbors as previously. So we can now start looking at what happens with the topology changes as this meristem, as this little leaf starts growing. So in the beginning, we have very few cells, as you can see here. We ignore the bordering cells because of contour conditions, because they're actually not neighboring much here. And so what we see here, for example, is we have a color coding, which is the number of neighbors that a cell has. So this white one has 6 neighbors. The browner tones here are for lower than 6 neighbors, so 5, 4, 3. So 3 if you're like a triangle. And up here we can get like cells which have 7, 8 to 9 neighbors, okay? So we see we have a certain distribution, right? And then as the leaf has grown very big, we see that we have this type of distribution here. We don't really see, there's something here happening in the midvein, but it's not really that we could see that there is some type of patterning in the neighborhoods that you have, okay? But we were interested in looking what happens to the frequency of those distributions. So how many have 6, how many have 5, how many have 4, how many have... If anybody has a question, ask about it. And the reason we were interested about this is because sometimes these things about, topology of a tissue, cellular material, it's a fingerprint about the type of biophysical constraints you're undergoing. What is it that you're trying to optimize? So cell surface minimizing, like bubbles, for example, right? Which have approximately the same area, right? They will go to 6, like a honeycomb and all that stuff, right? You guys are familiar. So let's take a look at those distributions. First of all, we are very aware that this is not a cell surface minimizing system because they become all wobbly. So we were wondering, hey, maybe it's going to give a very different distribution. So this is another, we looked at another leaf also just for confidence, which was not a mutant, right? And we see that as they develop, they very slowly go into this sort of characteristic profile here, where they have a certain preference here to have 4, 5 neighbors, and actually not 6. Yeah?
Yeah, that's very interesting, right? Because for Neumann's law, it's only 6.
Yes. Yes, so this is not for Neumann's law. There is something, of course, that there's a conservation in the, if you go more to 4 or 5, you still have to have a bigger breadth. Like the mean will still be somewhere. So, yeah. So here, for example, we look at a young leaf and an older leaf. And this is where we actually got a little bit, we thought this was, at least we got surprised. So this leaf here, these cells have undergone many more cell divisions, yet their distributions are right on top of each other. So it's as if in a very short time after, you know, a leaf is a leaf, it tries to preserve, or it is preserving, I'm not going to say try, it is preserving some type of distribution of how it distributes its number of neighbors. Okay, so this is totally data. We're not doing anything. We're just looking at data. We're just looking at numbers because we happen to have really cool data. You know, we were segmenting ourselves over time. We could follow each individual one. We had all these divisions, and we were just looking at it. Hey, you know, what happens? And, you know, then we thought, we were like discussing it. Oh, maybe the cells at the top, which aren't dividing anymore, but these ones are dividing, so maybe they have different distributions. So we look at the top cells and the bottom cells, and look, they still have very similar distributions, right? So they still have that peak here. In this case, they do have a peak at 6, but a shoulder at 5. So this isn't this. We compare this to other species. So we're looking at the species Arabidopsis leaf, so this one in blue. And we see that, yes, although it has its peak in 6, but it has a shoulder here at 5. And these other species have much more pronounced, like 6 frequencies. Okay, so then we make a model. Yeah, so this is a model. This is very reductionist. This is as reductionist that you can do. We make a model which we know is wrong, right? We forget about the shape. We forget about the biophysics of interaction. We say, you know what? We're just looking at a bunch of nodes which represent cells and their neighbors. And we're going to now have a simulation in which we let the whole tissue divide at the same time, or we pick one node and ask it to divide, pick another node and ask it to divide. So we can do this synchronously or asynchronously. Yeah? Okay. So if we pick a node, we can ask it to divide either in a way in which the daughter cell and the mother cell will end up with similar number of neighbors. Yeah? Or we can say it doesn't care at all. It just looks at any of its other edges and divides to one of them with equal probability, so totally random, right? Or we call this Pascal rule. There'll be like a favored preference to go to the middle, but there'll be some chance, which is slowing down, of it making an asymmetric cell division. Okay? So these are just rules, right? Okay. Let me tell you that there are lots of rules in biology about, oh, cells like to split in the middle, or cells like to split giving their, if you approximate them, like with the first moment, perpendicular to its major axis. There are many rules in biology about how cells split. But this has nothing to do with shape, okay? Just with topology. So we looked at what would happen in this in silico tissue, which is purely topological, which will have an equal split rule, Pascal, random, and we compare it to our data, the speechless. And really funny, this equal split rule that is looking at the number of nodes of its edges is on top of the speechless Arabidopsis leaf. And the others are not, okay. Now, as a side note here, we thought that was really funny because we definitely thought that the Pascal rule would be much better. It sounded more like, maybe sort of in the middle, but there'll be some type of variation. But the equal was really good at capturing this result. Now, does this mean this is what's happening? No. I just say that I use a model, I put a random rule in it, I get a distribution, I see that my data also gives me the distribution. It would be very incorrect now that we would say, oh, our cells, in reality, they divide in a way to keep the neighbors the same. But we can check the data, right? We can go back and check the data. So this is sorry for the numbers are a bit small. So this is the data looking at what's happening with the mother and daughter cell, a number of neighbors. So this is the experimental data where we actually followed 806 divisions. And in fact, when we compare them to what would happen to the random split, the Pascal, or the equal split, we see that it's actually very close to the equal split. So, okay, our simulation with the equal split gave something which looks like the wild type. When we look at how the wild type, the actual experiment is dividing, it is also seemingly following that rule. But is it really following that rule? Because now the question is, does a cell know how many neighbors it has, right? And these are the type of questions which are really interesting now. So, like, if I have a cell, well, first of all, if we have a cell which is elongated, we know that there are intracellular gradients where, suddenly, some things might like to position themselves like this, or some things might like to position themselves like this, or sometimes things might divide in the middle. So shape will have an influence on reaction diffusion things happening inside. But how can it know the number of cells? Yeah? So it'll try to always split in a way that the mother and the daughter are having the same. Could this be just a reaction to the shape, right? And so those are the questions. So then I'm not going to bore you guys to death. We also looked if shape would be the predictor, and it wasn't. So we could dissociate in the system shape with number of neighbors. So we're still stuck, and this is quite interesting. Might be, with this here, maybe because, we like to go camping in Wales. And, when we're camping, you always see this tension, where you put the pegs in the ground, right? And there is a lot happening where cells come in triple points, right? You know that there are sometimes even some molecules which concentrate there. Maybe there's some forces. So maybe it is not crazy that as a cell, you might not be, at least for this process, that aware of your shape, but you're actually aware of your pegs. And you're somehow dividing in a way to equilibrate your peg number. But this still would mean that we would start to have to look at the biomechanics, maybe at the cytoskeleton, at other things. And this is something where modeling is good for. It's not that we're saying this is how it happens, but it does make you think about something a little bit differently. And it's also very beautiful that we wouldn't have thought about this at all if we hadn't started looking at the data and just started to plot things. So it isn't always the case that you have to start a research with I have this hypothesis, right? Sometimes you can just look at something and then think, well, actually this begs a deeper question of how this might or might not be, built up. Okay, so just taking a step back, you guys will see a lot in this in the upcoming weeks on feedbacks, right? And so feedbacks is something which is really important for biology, right? And the problem is we have feedbacks on many, many levels. So you guys will be talking about ecology. So Johan will be talking, I think, about predator-prey also, right? But the reason I'm showing this is, again, this is one of those data sets which we happen to have because people were very interested in selling furs, right? Before, there was this thing against furs. And so they actually did know what was happening with the lynx population and the hare population. And so the idea here is that, you can see it in this peak at least, right? So we have the lynx and the hare population are very low here, but then the hares start going up, and so the lynx are very happy, they eat a lot, and then the hares go back down. So we have this predator-prey model, which you guys will be looking. But at the same time, there's a lot of noise, there's a lot of stuff that we cannot explain here. Right? Open questions. Is this because there are other top predators? Is it because, people would say naively, oh, maybe the climate, maybe something else happened? Is it because of spatial properties of how these things? It's not a well-mixed system. So it's always good to, when we have these models, to even think about very simple things, like if you have a spider and a mosquito. So this is one of the things that our biology students, they learn in year 2. They look at, if you have just a simple ODE, coupled ODE with spiders eating mosquitoes, what happens if you start spraying the mosquitoes or killing the spiders? And they find it always very counterintuitive that if you kill the spiders, you end up having more mosquitoes, but you don't reduce the number of spiders. So those are the type of feedbacks which you can only understand, not only just by looking at time plots of what happens here, and you could also, but you actually would have to look at this is as simple as you can write, I think, a predator-prey, mosquito and spider type of ODE. But it's very important now to look at, for example, phase planes, so nullclines, right? So this would be the nullcline of my mosquito. This is the nullcline of my spider. We see that we have an equilibrium here or here. You know, you can look at the vector field, and you can look at these trajectories, right? And you can see when do we get these cycling things or when do we not get them. So these are things I think that we will be developing, and certainly we will have some projects that you can develop, you know, looking at models, looking at the systems, dynamical systems properties of it, and trying maybe then to think about advancing these models maybe with stochasticity or with space, right? Because the pattern formation behind this is quite interesting. Another big thing in this area, which I don't think we will be talking too much in the upcoming 2 weeks, is fitting. So if you go out in the real life and say, oh, I like biology and physics, you're going to start getting data and people will ask you to fit for things. So we were working on something where we're looking at the different variants of COVID coming up and what's happening there. And you have to be very careful with fitting. Now, I know you guys probably heard this because my physics teachers were always telling me this here, right? Von Johann von Neumann, with 4 parameters, I can fit an elephant, and with 5, I can make him wiggle the truck, yeah? Now, it's true, but it's often said in a very negative way when you start doing biology because you will end up having lots of equations. And that's because there are lots of elements and there are these feedbacks. And it's not always good for you to reduce it before you understand what's really going on, right? So don't get told off that you have lots of parameters, because sometimes to do the mapping to something where you have a handle on experimentally, you will have to have lots of parameters. The problem is that you will have to actually do some type of dimensional reduction to be sure that you can understand the different regimes you can be in independently of those parameters, because you will not know what those parameters are. Almost certainly not really well, okay? And here I just want to sort of open up the talk so we can get a little bit more discussion. And that's the value of mismatch, okay? Probably you don't suffer this problem, but many people do. They want to have their model matching, really quantitatively get close to that point. And there's a big mistake here, right? And so thinking back, right, way back to Ptolemy and Ptolemy's view of the world, people laugh, like my kid was laughing at me, how stupid they thought things were going in circles. It's not stupid, because you can describe all those planets very well by circles on circles. It's not a problem. It's very beautiful, actually. You can describe them. You can actually predict when the next eclipse is going to happen. You can predict when you're going to have your alignment of your planets. You can predict a lot of things. So why was it wrong if you had predictive power and it was precise? Yeah? Why? Well, because you just overfitted, okay, obviously. And you wouldn't then be able to, in the mismatch of something happening wrong, you could, for example, if you do now take the standpoint that the sun is in the middle, right? So we're here, like in our truth. Then we can start predicting, for example, that Uranus was a little bit off, or that Uranus had to exist because Neptune was off, things like this. So when you're doing modeling, it is also important. So on one hand, I say, yes, you have to have the mapping to the experiment. But on the other hand, I'm also trying to tell you, it's okay to be approximate or to be wrong, right? And then you can actually contemplate in what way am I wrong and what else is needed, or can I now understand, well, this simple model would never give me this. I could put some noise on top of it, but it's essentially needing some type of other process, right? So be aware, because as a modeler, we somehow unconsciously think that our model wins or it loses, right? And what's the quote? Every model is wrong, just some are useful, yeah? And so mismatch is not something to run away of. So I have a couple of minutes. I'll just show this work from Stan Murray and Leah Cachette, which, to give you an example of that. So you guys heard of type 1 diabetes, so when kids get diabetes, like young. It's because when children are born, their pancreas, they have to reform these islets of Langerhans, and these are the places that will produce insulin, right? So when you're reforming them, there's a lot of these, because you have a lot of it around, you have a lot of apoptosis, a lot of these cells, these beta cells, they'll die. And then you have the macrophages coming in, which are going to eat them up. They have to clear them from the system. If the macrophages fail to clear up these beta cells, you're going to get autoimmune responses. Autoimmune responses that are going to start destroying all of your beta cells, and then you don't have anything to produce insulin. So then you have the diabetes. But so they knew a bit about this, and in vitro, you can take now these cells out from a mouse which is healthy and a mouse which has type 1 diabetes. And so the pink blobs are your macrophages, and the black things are your beta cells being eaten up. So this is the type of data they had. Okay. And now they want to understand what is the process which leads some cases to form healthy individuals and others the type 1 diabetes. So you have a mathematical model. So this will all be like ordinary differential equations, right? Mass kinetics. So you have your macrophage in the beginning, it's M0. Then it starts, eating these apoptotic cells, those dying beta cells. If it has 1 in it, it's M1. If it has 2 in it, it's M2, and 3, M3. So you can actually count them, right? But now they hypothesized, because they didn't know the mechanism of this. So how quickly does it start up slowly, or does it start up without a back reaction, and then here you activate macrophages, so you have a persistent memory of activated macrophages. So that would help you clear up later. So you have different, very complicated models. And now you have your model splines here. So this is M0, M1, M2, M3. So those are your populations depending on how many beta cells they have engulfed. And these data points here are data points, right? So you're looking at, you killed a bunch of mice here and a bunch of mice here, healthy mice and your diabetes mice, right? And you're counting, right? And you're putting them together in vitro, these things, and you're looking what's happening. So it fits very well. And now you have different model variants, right? So what is the best model? Okay, now this is a question which will come up a lot. Okay, so we like to believe in physics that, it's the one which is most parsimonious, right? Because somehow, things parsimonious seem more true, right? That's what lots of physicists say. It could be the best fitting model, so that we make sure that our model fits the best to every single point. It could be the one with the strongest predictive power, that I just do it until 4 days, and then after that I see, does it get to 6 and 8 days correctly? So do you have this extrapolation power? Could be. It could be that one model, it needs to have that auto-activated state, so I can tell to the experimentalist, let's take a look for that other state. Is there some way now with a quantitative reasoning to go back to the experiment and devise it in a way that I can catch something or not? So that's going from the quantitative to the qualitative, okay? So that you see something. Or you can look at what model is most likely to best capture the data. And this is not the best fitting one. It's the most likely to capture the data. So one thing that helped me a lot, because I didn't see this in physics, I'm not sure about you, ICTP-SAIFR information criteria. So basically, if you have 2 models that you're working with, you can compare both of them, right? And you can say, which of them is closest to the model out there which would best describe it? I don't know even what model that is, but I can ask the question, is A or B closest to my ideal model? And I find that quite magical, and it's very beautiful, because in this case, you don't have to worry about the nitty-gritty of everything, but when they look at the ICTP, so it gives you a number here, right? So the lowest number is the one which is closest to your ideal model, which you don't know what it is, but at least you know you're closer, right? And so you see that here for your mutant, the NOD mice, the mutant mice, you see that the basic model is really close. So that suggests that in the NOD mice, what's happening is that you don't have your auto-amplification of your macrophages. Whereas in your wild-type mice, you could see here that the one with the best predictive value, here 38, here 835, was the one which you do have this replication included. Okay, so that is a way of working. I don't think we're going to do too much of this, but it's just to know that everything you're doing here with modeling, at a certain moment, there might be some data, and there are ways now of interacting with the data in interesting ways. So when I ask my students, which are biologists, why do you do systems biology? Why do you think it's important to have models? They say it's to predict things, like predicting the weather. Yeah? And yes, that is one reason why someone could do modeling. So I'm not going to talk about Bayesian here. But I would say another thing to do, and this is work that we do, is actually to use it when you have a big netball of interactions to try to predict novel outcomes, things that mismatch. Because then you know, okay, I took something I knew, I tried it out, and haha, something's happening that's not happening in my experiment. Okay? So then you can go back, and I can talk about this. Hypothesis generation. Sometimes, I hope I can give you some hands-on experience on this. Sometimes we look at cellular mechanics, so cells interacting through adhesion, right, moving around and all that. And we can get hypotheses, for example, of why when dictyostelium is moving through the soil, cells aggregate or disaggregate. And what was shown historically is many of these models came with a hypothesis even before the predictions, the experiments could be made. So this is a science paper which actually used work which was done in theoretical biology to be able to understand what was happening. So it generates hypotheses. You don't even have to work with data sometimes. Sometimes you just show, hey, this is possible. This mechanism could give you this type of cell sorting. Another thing is explanatory power. So going back to Dennis Noble, one thing in pattern formation that we will talk about is excitable media, right? So what they are, how you can get them. Excitable media are very much described to understand the propagation of an action potential through your neuron, right? But the nice thing is, if you take all those equations of like neuron and neuron activity and you reduce them down to Nakumo's simple models, you can actually start explaining a huge set of phenomena that happens in biology. Not just your neuron propagation, but you can understand, for example, why once you get something called spiral breakup, your spirals form on a heart, and that can happen because some regions are a little bit more heterogeneous, right? So this is something that happens by itself, why you could get fibrillation. And it is interesting to note here that it was theoretical biologists that proposed that why you get these arrhythmias or why you can get a heart attack, right, is because of your wave, your excitable media wave, suddenly forming a spiral which auto-sustains itself, yeah? And so what you can do to diminish this is knocking someone's heart or giving it an electric pulse, right? Because then you try to reset that whole state. And it was laughed at, right? People would laugh at that until it was experimentally seen that you could see a spiral on the heart of a dog, right? And nowadays, it's the way that we go about understanding a bunch of diseases in the heart. Yeah? But it's not that we knew enough about the heart to be able to write down these equations. We had generalized equations from a different field, knowing that they could give us excitable media, and excitable media has the property to give us spirals that we could start seeing and predicting that spirals would happen in different systems. So this is just a bunch of different systems from like oocytes to cyclic AMP to the heart, right? In which we see the same type of principles. So that's why pattern formation is so important, right? So once you understand it for one mechanism, like to try to understand what are the key components that form that pattern, and then you can have this thing called isomorphism. You can start seeing these properties in other systems biologically. Biologists have a problem with this, right? Because they think very much in terms of evolution. So if I have a mechanism in plants, they don't understand how I could use that in animals. But it's about the mechanisms, about the pattern formation, the dynamical systems process that we can see there. Yeah, so this is what I want to say. So we can predict beyond the system under study. Another thing which is really important with systems biology, I think we will do here is, and I'm going 3 minutes over time, is communication, right? So even I think physicists, like, you go from solid state physics, someone else from nuclear physics, you might be talking about the same thing, but using different terminology. Now think how vastly different this is when you start talking to biologists or biomedical people, use their little jargon. So by communicating together, it is really important because you start understanding that you're talking about the same thing, but possibly in different ways. Okay? And lastly, I hope that we can get to this, especially in the last week, why it's fundamental to do systems biology is because we cross scales, right? So it's not that we can just look in the cell and that's it, right? The cell is interacting with the tissue with the cell. So those type of feedbacks which cross scales makes it inherently difficult to use just one model formalism for everything, right? Also temporal scales, right? You can have a process which is changing like very slowly, like on days and weeks, but it is still able to have some type of responses on the order of seconds, right? So how do we cross those type of scales in a modeling formalism? It's probably not going to be pretty, not going to be simple, not going to be small, but how do we do that we can still logically follow the steps? So those are maybe a little bit more challenges, but I think you'll be getting a bit of taste of all of this during the next weeks. And yeah, I think the floor is of you guys now, if there's any comments or anything you'd like to point out at all. Okay, so thanks for that.
Is there any question?
Yeah.
Hey, good morning. Thank you so much for presentation. First of all, really loved your presentation because, you know, I'm a picture guy, so I really love to see a lot of pictures and stuff. But what I was about to ask is, like, when you were talking about the ideal biological model, you say that if you have two models, you can, like, kind of compare them and use them to see if they are close to one ideal model, and then you come up with some numbers. My question is, like, how do you come up with the numbers if you don't know what the ideal model is?
Yeah. That's really good, because we always think we would have to know that to subtract, right? So because you have your data points and you would, so this idealized model isn't even something that you would write down, but it takes, basically what it takes into account is, there is a threshold here for, like, simplicity, so you don't want to have too many, you don't want to overfit, but you still want to be close enough to your data points. So you could say it's a mixture of that, but it's also, this ideal model is also somehow thinking what would be the best that you could accomplish using those elements, right? But I can show you a little bit later how that goes, but I know it does sound, so it's like implicit type of, the application theory. Does anybody use it? Yeah. Is there a better way that you would describe the ideal model? It's just that, yeah, it's.
If you make, so you have a stepwise approach where you compare the different model versions, you have an increase of the complexity of your model, and that ICTP information criteria poses a penalty on the use of more variables. Basically, so saying, okay, you have added one variable, but then you only gained a little bit of better information. So is it worthwhile? And it proposes a trade-off for you to look at, is this really a better model given the increase of predictive power? Maybe it's just a minute increase of the fitting. Is it worthwhile to have a more complex model then? And that's what it's all about. I must say, it's a long time ago that I used it, but that was the setting that we used it.
Exactly, but you do have to compare between models that you have, right? So you don't compare it absolutely. And it's not a blind thing that I say, look, I don't allow, because it could be, in fact, that you will have lots of new parameters, which it won't put the penalty in, because you are getting your quality of result, right? Yeah.
So, is there any more questions?
Just a question, does someone have, do you have a question? Yeah.
Okay, thank you for the talk. I was curious about the cell division problem.
What problem?
The cell division one, I would have thought if you are growing while filling the area, shape would be very important. Like if you have squares or if you have triangles, the number of neighbors would change a lot. So I was curious whether you thought about this or...
Yeah. So indeed. So that's the first thing. I mean, space is important because it's a space-filling problem, and so one thing we did, we tackled this in two different ways. One is we did look at the data, and we saw that you could have a smaller daughter cell and a bigger mother cell, right? So in this system. In other systems, it's very much cell area. So this is by no means like a universal cell division rule, okay? So that is one thing. But it's a little bit unsatisfactory because, it could be cell shape. There is also some studies where they considered the tension through biophysics, and they got similar results. So what I want to say is maybe another hypothesis could give a similar result. We see it only in terms of neighbors, but it might also have a correspondence on some type of biophysical property. It happens to have. Now the question is, what does the cell feel? Yeah. So what is being read out by a cell? And by the way, maybe I'm going astray, but very often it is very tempting for us to put physics, like, oh, the stresses are greater here, so the cell does something because of stress. But how does a cell measure stress, okay? And yes, there are a lot of people trying to look at that, but it's actually quite it's not that well known, the processes at which stress can be read out. Also, it's very difficult to know the direction at which a force is being applied. Like you might know that I'm being squeezed, but am I from above or below? So all of these things, although it's easy for us as the looker to imagine that being the rule, sometimes you have to put yourself in the mind of a cell, and that's really crazy because a cell doesn't have a mind, and think, okay, but what is it seeing, and how would it be able to translate something that I see as a stress in something which is either molecules moving around, or a pattern being formed, or some type of tensegrity in your cytoskeleton. Yeah. So that's why I would still say that area, it's not in this case, but it could still be something mechanical we haven't considered, but then it would have an overlap with this thing. So that's why it's interesting now to go to the molecular things. And that's where mutants are really interesting, because you might find a mutant which does it differently, and then you can ask, but what is the coding, and what part, is it in the cell polarity mechanism? Is it in the, and then it gets interesting because we know the correct mapping, right, to that. Yeah, good question. Yeah, Erin?
Yeah, wonderful talk, putting things in perspective. I liked it. One thing I find myself constantly torn between is this, trying to quantitatively model an existing biological pattern or phenomenon that I see. And the other is to kind of, take this as a given, but focus on the principles and the mechanisms and the physics behind that. And I think you said it, but as a physicist, I tend to lean more towards the mechanisms and the principles, because maybe, as you say, they are more generally valid and can be applied in a more broader way. So as somebody, that's my question now, in mathematical biology, how do you feel this tension? How is that for you?
Yeah. So this is a really good question. So first of all, what you're doing is amazingly important, right? Like to look at the principles of the pattern itself and the mechanism, but the problem is a misunderstanding of how you communicate, not you, but how that community communicates that down to the biologist, and how the biologist understands what you are saying, right? So when you write down a system, you say, this system can give me these type of phenomena, right? The biologist, he's looking for a thing, not a process, not a feedback, not an equilibrium state. He's looking for, I want a protein. I want another protein. I want something, right? He's looking for something very concrete. And he might miss a step that there might be some levels in between that and what you're describing. So, right? And to fill in those gaps, I think that's where the communication is important, because, so for example, historically, everyone is touching something that happened in the past in theoretical biology, which gave theoretical biology a bad name for biologists, is, for example, Turing patterns, right? So Turing patterns, they can form stripes, and then some people said, oh, I see stripes in my system here. So that means I need an activator, I need an inhibitor, because you told me it's an activator and an inhibitor. You wrote down an A and you wrote down an I. And it gets even worse, this field, because the activator, they say, oh, the activator is something then that is activating itself, inhibitor is inhibiting the activator, which isn't even the way the Turing had written it, right? Okay, but so they search for something which, like zebrafish, right? Zebrafish, it is an activator inhibitor mechanism, but they see it has to do with cell dynamics. So something can reinforce itself, that something can inhibit itself. It's going through a different mechanism, but you can still write it as activator inhibitor, and you can still make predictions of how the stripes are changing, but you won't find those variables as concentrations of something, right? And I think this is where I think maybe every case, at the moment, every case is a new case for us to discuss. But the question, first of all, is the system really what you think it is? That's one. Because, for example, what was it? The Drosophila lines, they were lines, but they weren't Turing lines. Yeah? So that's one thing. And a dynamical systems approach could have shown her probably it's not going to be Turing lines because they don't behave as such. So you can look at the symptom. You can even, so maybe you shouldn't even look for an activator inhibitor to begin with, because that's not what would be predicted. But then there might be other systems, it behaves in every sense of the way for perturbations for this, like that dynamical model class. And then you can start thinking, what are the things that would be necessary? You do need to have some spatial coupling here, which is larger than that spatial coupling. And we're not saying that it's going to be a smaller molecule, but something has to be. It could even be a force, right? It could be something else. And that's where you have to start, thinking together of how to do the mapping, right? So it is super important that our biologists, experimentalists, understands the possibilities of dynamics, so they understand even how they can make experiments they wouldn't have thought of doing, because it's not about the things, it's about the process. And once they categorize like a disease, right? Disease is negative, but once I have a diagnosis, okay, it's this, right? So I see these hallmarks. Now I can start digging in and start thinking how it is imprinted, how it is designed, right? But that narrative is often lacking, and that's why people say, okay, he was wrong, he put me on the wrong foot, this is way too simple, I have more things happening. You're going to get this type of rhetoric. I sound very political here, but it is a bottleneck of doing science, is sometimes misunderstanding what it is that your model is showing and what it is not showing, right? So, we like to say sometimes what it is that we're doing, but we forget to say what it is that we are not doing, and that could be just as important, right? What you are ignoring or what you are not saying about the system. Yeah. A long answer, but there we go. Okay. So I'll be around. We can have more discussions and yeah.
Yeah. So I was going to suggest that, and thank you again, Veronica, for the great talk.
Thanks.